Thanks so much for the introduction. So as the final speaker before the break, I promise I'll try to keep this short. So my name is Pamela Gan, and I am from NalaGenetics, which is a startup based in Singapore. And today I'll be talking about our experience using adaptive sampling for pharmacogenomics, as well as genome-wide variant analysis. So I think many of this audience is familiar with the concept of pharmacogenomics, or PGX. Pharmacogenomics investigates how genetic variations can affect our drug metabolism, efficacy, and safety, with the idea that not all drug treatments should be given equally, but should be tailored according to an individual's ability to metabolize those drugs. And one factor that influences this is our genetics. Some of the challenges that still face this field is that some PGX genes are highly polymorphic. They are found in complex genomic regions, so they're found close to pseudogenes, which means there's a lot of structural variations, duplications, deletions, as well as hybrid formation. And also, there are rare and novel alleles present in the population, as well as, importantly, the need to call combinations of alleles or star alleles or haplotypes rather than just single variations themselves. So for example, for these individuals in the bottom right-hand corner, even though they both have, they're both heterozygous for the same two variants, they both have different metabolic profiles because the combination of the variants are different. So to this end, we developed this end-to-end workflow for PGX profiling using Oxford Nanopore's adaptive sampling technology. And we validated this workflow with 10 experimental runs across 30 samples. So it's run in 3-plex format using different sample types, including reference cell lines, external quality assessment cells, as well as clinical research samples that have been validated orthogonally with microarray as per the current standard of clinical testing in Singapore. And some of the key outcomes from these experiments are that we achieve, across the 10 runs that we ran, we achieve a mean on-target coverage of 25.2x and a mean off-target coverage of 3x, which means an enrichment factor of about 8.5 times. And we tested both SUP and hack-based calling and found that, as it can be expected, SUP-based calling provided very high accurate calls for small nucleotide variations and also achieved high concordance above 95% for structural variation calls. And how does all this translate into the final data, or rather the diplotype calls for these genes? So here we look at 11 of the 20 genes of interest, because these are the ones with diplotype calls. And where you see the light blue is where it's concordant for the with the reference data set. Where it's dark blue is where upon manual inspection, we can see that it's improved call. And where it's red is where it's discordant. So in the majority of cases, we can see that the adaptive sampling protocol achieves either concordant or improved calls compared to the reference data set, with less than 1% having a discordant call. And in many cases in general, the improvements in calling were based due to better phasing and expanded haplotype coverage, as you can see in this sample here, where in the GetRM reference data set, this was not able to be resolved. So there were 2 potential combinations of diplotypes in this case. And when we ran short read type tools for this diplotype calling, they could phase it to different levels of success. With adaptive sampling, we could conclusively get a resolved call and very strongly supported by the read data, as you can see here. And we did see some discordant calls, and in this case, this was due to lower variant calling performance caused by allelic imbalance. But I'll note that these samples were, or these regions rather, had lower than average coverage, so 15x in this sample here. And finally, we also looked at the off-target reads and found, as you can see here, the light blue and the blue. These are the adaptive sampling, the performance of the adaptive sampling in terms of imputation performance. And it's very similar to what can be achieved with short read low pass sequencing at the same depth. And in both cases, these are superior compared to the microarray genome-wide. So this is the summary slide, but I'll say, because I'm running out of time, that basically we think this is a very promising first step to implementing this in clinical practice. And finally, I'd like to thank all the people who have worked on this project, so the team at NalaGenetics and our collaborators at Tan Tock Seng Hospital and National University Hospital in Singapore, as well as the funding agency. So, and also to you all for your attention, and I'm happy to take any questions.